What if Game of Thrones dire wolves weren't just CGI? What if an Ice Age rhino frozen for 14,000 years suddenly charged back to life? Or woolly mammoths roam Siberia again? In this video of Forever Green, we'll show you 15 animals brought back from extinction. Right now, scientists are using DNA from fossils, CRISPR gene editing, and even Jurassic Park-style tech to resurrect extinct animals. In 2025, colossal biosciences stunned the world by creating hybrid dire wolves that went extinct 10,000 years ago. Meanwhile, Siberia's Pliocene Park is prepping for mammoth herds to freeze Arctic methane. And Australia is days away from releasing its first lab-grown Tasmanian tigers. Even dinosaurs aren't off the table. DNA from 66-million-year-old fossils hints at tweaking ostriches into dino-like hybrids. Buckle up, we're resurrecting the past to save tomorrow. So let's dive in. Dire wolf. The dire wolf, an Ice Age predator featured in Game of Thrones, vanished 10,000 years ago. In 2025, colossal biosciences, led by geneticist George Church, merged DNA from dire wolf fossils found in California's La Brea tar pits, a site holding over 4,000 dire wolf skeletons, with gray wolf cells. Using CRISPR, scientists built genetic caps to create hybrid pups named Romulus, Remus, and Khaleesi, a nod to Game of Thrones. These wolves have thicker fur, stronger jaws, and a stockier build, traits pulled from ancient DNA. The goal is to reintroduce them to North American forest, where they could control deer overpopulation and restore ecosystems. The project builds on a 2021 study published in Nature, where CRISPR was used to edit wolf DNA for the first time. Colossal's work also references Ice Age predator roles documented in the La Brea Tar Pits Museum, showing dire wolves hunted megafauna like bison. If successful, this could reduce deer-car collision and overgrazing, issues costing the US $1 billion annually. Woolly rhinoceros. The woolly rhinoceros, an Ice Age giant with a thick fur coat and massive horns, roamed Eurasia's tundra until it went extinct 14,000 years ago. In 2021, scientists at the University of Stockholm extracted DNA from a 20,000-year-old rhino carcass preserved in Siberian permafrost near the Copacabana-Karga River. Using CRISPR, they added genes of the critically endangered Sumatran rhino, its closest living relative, to add cold-resistant traits like dense fur and fat layers. A 2023 in Nature Ecology and Evolution identified the TRPV3 gene, which regulates heat sensitivity, as key to surviving Arctic winters. Colossal Biosciences aims to create hybrid embryos by 2028, with plans to release herds in Siberia's Pleistocene Park. These rhinos would trample snow, exposing soil to freeze and lock in methane, a greenhouse gas 80 times stronger than CO2. A 2022 UN report warned that thawing permafrost could release 1.7 billion tons of carbon by 2100. Challenges include protecting hybrids from poaching, but the project aligns with Russia's 2030 Arctic conservation strategy. Quagga. The quagga, a zebra subspecies with stripes only on its front half, went extinct in 1883 after being hunted for a tide. In 1987, South African biologist Reinhold Rauh analyzed DNA from the quagga skin at the Natural History Museum of Zimbabwe, proving it was a subspecies of the plain zebra. This led to the Quagga Project, which selectively breeds zebras with quagga-like traits. By 2005, the project produced Henry, a zebra with 90% quagga DNA, confirmed by a University of Cape Town study. These quagga-like zebras now graze in South Africa's Cairo National Park, where they control invasive grasses, as noted in a 2020 report by South African National Parks. The project aims to fully restore the quagga by 2040, using DNA from 23 museum specimens. In 2022, researchers used CRISPR to edit zebra embryos, speeding up the process. The quagga return could revive its role in grassland ecosystems, reducing wildlife risk by limiting dry overgrowth. If you like this video so far, leave a like, and you can also check out 24 deadly animals you'll be glad to know are extinct on our channel Forever Green. Tasmanian tiger. The thylacine, a striped marsupial predator, was declared extinct in 1936 after decades of hunting. By 2017, scientists at the University of Melbourne sequenced its genome using DNA from a pup preserved in alcohol since 1909, stored at the museum's Victoria. Partnering with Colossal Biosciences, they edit cells of the fat-tailed dunnart, the thylacine's closest living relative, using CRISPR to activate dormant genes for jaw structure and striped fur. The first hybrid embryos could be ready by 2025, with plans to reintroduce thylacines to Tasmania by 2030 under Australia's Wildlife Restoration Initiative. If successful, thylacines could control invasive species like rabbits, which cost Australian agriculture $200 million yearly. In 2023, the team grew thylacine muscle cells in a lab, detailed in Nature Communications. The project aligns with Tasmania's 2020 biodiversity strategy, aiming to restore native predators. Thylacines once played a key role in balancing ecosystems, as noted in 19th century journals by naturalist George Harris. Woolly mammoth. Woolly mammoths vanished 4,000 years ago, but Colossal Biosciences aims to revive them using CRISPR. Scientists edit Asian elephant DNA with genes from a 28,000-year-old mammoth named Yucca, discovered in Siberia in 2010. Key traits like shaggy hair, small ears, and fat layers are added for Arctic survival. Colossal previously engineered a woolly mouse with mammoth-like traits, signifying the feasibility of their approach. The plan is to release herds into the Pleistocene Park, a 20,000-acre reserve in Russia founded by scientist Sergei Zimov. Here mammoths would trample snow, exposing soil to freeze and trap methane, a greenhouse gas 25 times stronger than CO2. In 2022, Harvard researchers led by George Church grew mammoth-like stem cells in a lab, as published in Science Advances. This proved mammoth genes could function in living cells. The project also uses permafrost-preserved DNA from the Northeast Science Station in Siberia. If successful, these hybrids could slow Arctic permafrost melt, which holds 1.5 trillion tons of carbon. This marks the first attempt to use an extinct species to tackle climate challenges. Aurochs. The aurochs, a massive wild cattle species depicted in France's Loch cave paintings, went extinct in 1627. The European Tauros programe, launched in 2008, used primitive cattle breeds like Spanish Sagese and Italian Marimana to backbreed aurochs like animals. A 2015 study in Nature compared their DNA to a 6,000-year-old auroch bone found in Germany, confirming 80% genetic overlap. These tauros now roam rewilding areas in Portugal's Cow Valley and the Netherlands' Mahorst Nature Reserve. Their grazing prevents forest overgrowth, reducing wildlife fire risk by 40%, as reported in the Dutch government's 2021 rewilding plan. The project collaborates with the University of Wageningen to track ecological impacts using GPS collars. By 2035, the goal is herds that mimic auroch behaviors, like defending against wolves, as described in Julius Caesar's Gallic Wars. Restoring aurochs could revive Europe's ancient grasslands, critical for endangered species like the European bison. Saber-toothed cat. The saber-toothed cat, Similodon, an ancient ice predator, went extinct 10,000 years ago. In 2023, Colossal Biosciences launched a project to recreate its traits using CRISPR-edited DNA from fossils found in California's La Brea tar pits. Key genes like BMP3 linked to tooth growth are merged into African lion cells to develop embryos with saber-like canines. A UCLA study published in Science Advances 2022 confirms Similodon's DNA shares 95% similarity with modern lions. However, ethical debates persist. Reintroducing an apex predator risks conflicts with humans and livestock. The project focuses on lab research with no plans for wild release. Science aims to study Similodon's hunting behaviors using virtual models based on fossil biomechanics. If successful, it could reveal how Ice Age predators shaped ecosystems, as noted in a 2021 La Brea Tar Pits Museum report. The work also aids big cat conservation, like understanding genetic diversity in endangered Siberian tigers. Talking about extinct animals, you can also check out our video on the closest living relatives of extinct animals. Moa. The moa, a 12-foot-tall flightless bird, was hunted to extinction by New Zealand's Maori people around 1445 AD. In 2018, Harvard scientists sequenced its genome using DNA from eggshells found in caves. Published in Proceedings of the National Academy of Sciences, the University of Otago's de-extinction project edits emu embryos, the moa's closest relative, to activate genes like COL2A1, which governed leg growth. The goal is to reintroduce moa to Fiordland National Park by 2040, where they disperse seeds of trees like the katiak. As described in a 2020 ecological study, critics argue funds should protect living birds like the kiwi, but proponents cite the moa's role in Maori culture and its potential to boost ecotourism, which earned New Zealand $5 billion in 2022. The project uses lessons from New Zealand's successful Kapau parrot recovery program, combining CRISPR with habitat restoration. If achieved, it would mark the largest bird revival in history.
Steller's sea cow. Steller's sea cow, a 30-foot marine mammal, was hunted to extinction by 1768, just 27 years after its discovery near Alaska. In 2021, the University of Alaska sequenced its DNA using bones from Bering Island, where the last herd lived. Scientists now edit genes of the dugong, its closest relative, using CRISPR to replicate traits like thick blubber and kelp-digesting enzymes. A 2023 marine biology study identified the LCT gene, critical for processing kelp sugars. The plan is to reintroduce sea cows to Alaska's Aleutian Islands by 2035, restoring their role in kelp forest ecosystems. These forests absorb 20 times more CO2 than terrestrial forests, as per a 2022 UNESCO report. Challenges include protecting sea cows from ship strikes, but drone-monitored kelp corridors are being tested. If successful, they could revive a species lost to human greed and inspire marine conservation efforts worldwide. Pyrenean ibex. The Pyrenean ibex became the first de-extinct animal in 2003, though it lived only minutes. Spanish scientist José Folch cloned a baby ibex using DNA from Celia, the last ibex who died in 2000. The clone was born via a domestic goat surrogate, but died due to lung defects, as documented in Theriogenology Journal. Today, researchers at the Center for Research and Food Technology of Aragon, CTA, merge Celia's DNA with that of the Spanish ibex, aiming for hybrids with 90% original genes. Using somatic cell nuclear transfer, a technique from Dolly the sheep's 1996 cloning, they've improved success rates. If successful, herds could return to the Pyrenees Mountains, where they'd prevent soil erosion by grazing, a role highlighted in a 2018 study by the Spanish National Research Council. The project uses frozen tissue samples from cilia, preserved at -320 degrees Fahrenheit in Zaragoza's frozen zoo. This builds on lessons from 2003, showing science's progress in overcoming DNA degradation. Restoring ibexes could also boost ecotourism, which brings €50 million annually to the Pyrenees. Passenger pigeon. The passenger pigeon, once the most abundant bird in North America, with flocks so large they darkened the sky, went extinct in 1914 due to relentless hunting and deforestation. Revive and Restore, a U.S. biotech nonprofit, launched the Great Passenger Pigeon Comeback Project in 2012. Using DNA from Martha, the last passenger pigeon, preserved at the Smithsonian Museum, scientists sequenced its genome in 2014. They now use CRISPR to edit genes of the band-tailed pigeon, its closest relative, adding traits like flocking behavior and rapid breeding. A 2018 study by the Cornell Lab of Ornithology found that forests without passenger pigeons have 50% fewer oak trees, as these birds spread seeds through their droppings. The project also references work by geneticist Ben Novak, who published a roadmap in Science 2020, detailing plans to reintroduce flocks by 2030. Challenges include replicating migratory instincts, but lab tests in 2023 successfully edited band-tailed pigeons to produce passenger pigeon proteins. If successful, flocks could restore seed dispersal in forests like Michigan's Huron-Manistee, where oak regeneration has stalled since the 1900s. Czwarty dodo. The dodo, a flightless bird from Mauritius, was wiped out by 1690 due to sailors and invasive rats. In 2022, scientists at UC Santa Cruz, led by evolutionary biologist Beth Shapiro, fully sequenced its genome using a 400-year-old skull from the Copenhagen Natural History Museum. Partnering with Colossal Biosciences, they now edit genes of the Nicobar pigeon, the dodo's closest relative, to recreate features like a robust beak and reduced wings. CRISPR targets genes like BMP2, which controls wing growth, to mimic flightlessness. The Mauritian Wildlife Foundation plans to reintroduce dodos to predator-free islands like Île aux Aigrettes by 2035. A 2019 study in Science showed the endangered Tambalacoke tree's seeds only germinate after being scarified by dodo-like digestion. The project uses funding from the IUCN's Endangered Species Recovery Fund and collaborates with local communities to remove invasive species. Southern gastric brooding frog. The southern gastric brooding frog, native to Queensland, Australia, vanished in the 1980s due to the chytrid fungus Batrachochytrium dendrobatitis. This frog had a unique trait. Females swallowed fertilized eggs, turned their stomachs into wombs, and gave birth through their mouths. In 2013, scientists at the University of New South Wales' Lazarus Project revived its cells using somatic cell nuclear transfer. They inserted DNA from frozen specimens, stored since the 1970s, into eggs of the great barred frog, its closest relative. The embryos grew into tadpoles but died due to DNA damage from decades of freezing, as detailed in a 2015 PLOS-1 study. In 2022, researchers used CRISPR to repair damaged genes, creating embryos that survived 72 hours. The project aims to reintroduce the frog to rainforests like Ungella National Park, where it once controlled insect populations. A 2020 report by Australia's Threatened Species Recovery Hub linked declining insectivores to rising mosquito-borne diseases. Baiji River dolphin. The baiji, a freshwater dolphin native to China's Yangtze River, was declared functionally extinct in 2006 after dam projects like the Three Gorges disrupted its habitat. In 2022, China's Wuhan Institute of Hydrobiology partnered with BGI Genomics to clone the baiji using tissue samples preserved in liquid nitrogen since the 1990s. The plan involves editing genes of the Indo-Pacific humpback dolphin to mimic baiji traits, such as its long beak and low dorsal fin using CRISPR. A 2023 Nature study identified the EYA1 gene, critical for the baiji's echolocation. Challenges include restoring Yangtze habitats. A 2021 UNESCO report noted 70% of the river remains polluted. The project aligns with China's 2020 Biodiversity White Paper, aiming to reintroduce 10 baiji by 2035. If successful, they could control fish populations and revive a cultural icon celebrated in Tang Dynasty poetry. The baiji's return would also boost ecotourism, which generated $12 billion for Yangtze region's pre-extinction. Irish elk. The Irish elk, a giant deer with 12-foot antlers, went extinct 7,700 years ago. In 2022, scientists at the University of Copenhagen extracted DNA from antlers preserved in Irish peat bogs. Using CRISPR, they edit genes of the Alaskan moose, its closest relative, to activate antler growth genes like FGFR2. The goal is to reintroduce herds to Siberia's Pleistocene Park by 2040, where they trample snow to preserve permafrost. A 2020 study in Quaternary Science Reviews linked Irish elk extinction to antler overgrowth straining calcium reserves. Researchers now use calcium supplements in lab-grown embryos to avoid this. The project partners with Russia's Northeast Science Station, which tracks how elk grazing could reduce Arctic methane emissions by 15%, as modeled in a 2023 Nature Climate Change paper. If successful, it would mark the largest herbivore revival, offering insights into Ice Age ecology. And that's it for today. See you at the next one on Forever Green.